Okay, let me introduce the next speaker, Dr. Deeksha-Bhartiya. She is an entrepreneur and founder of Genomic Solutions, a young startup that aims to provide bioinformatics data analysis services. Dr. Bhartiya completed her PhD from the Institute of Genomics and Integrative Biology, Delhi, and she has a research experience over a decade in genome informatics and has expertise in analyzing and interpreting next generation sequencing datasets. So welcome, let us welcome the speaker and yeah, over to you.
This talk is meant for early career research people and to motivate young PhD students about through our journey. And thank you for the introduction, and thank you Karthik and Arpita for inviting me. I am thinking myself as an outlier in this conference because I am nowhere from the computing space, though I come from biocomputing area. So basically, I will be talking about the applications, mostly the applications of all the algorithms, the theories that you are building in the biologic field, and especially how you can use that, all this information for patient care. So I have done my master's and PhD in the field called bioinformatics, wherein we used to analyze, we actually analyze a large high throughput biological data sets, especially genomic data sets. So when I was talking about this genomics during lunch, a student asked me, okay, what is exactly a genome? So I realized that I have to start from there, that, okay, if I have to talk to an audience where people don't know anything about genome or genes, then I think I should start from there. So, on top of it, we are working on the mutations in the genomes. So since yesterday, I'm very sick here, and I was having this joke everywhere around that because I come from Delhi, I think my genome is mutated, and I can no longer bear clean air. And since I landed here, I'm having fever and cold. So sorry for my hoarse voice. But when I say mutations, then you should also understand that all of us, so the genome that we have in the cell, there are 3 billion base pairs in our genome. So yeah, so our genome consists of 3 billion base pairs, and if change in one single base pair of those 3 billion letters of the DNA that we have can cause a very severe disease such as cancer. And if we can accurately identify that mutation or the change in those 3 billion letters, we can not just help in diagnosing that disease, but also provide way for future treatments. And this entire process is called genome testing, wherein we use that information that what particular letter in the DNA has changed in a patient, and how can we use that particular information to do that analysis. And since I've started to giving you the perspective, let me introduce my startup called Genomic. Genomic is actually the Hindi word for genomics, and we started in late 2021, right during my postdoc. So I did my PhD at IGIB. There's an institute in Delhi, CSR Institute. After my PhD, I thought that, okay, I'm nowhere less than publishing high quality articles, and I'll definitely get a Nobel Prize. And I landed up in a government job in ICMR, and I realized that, okay, I need to do something else. This is not where I belong. And after spending around 3.5 years as a bioinformatics scientist in an ICMR institute, working extensively though on cancer genomics, I wanted to get far more exposure, and I went for postdoc at Karolinska Institute, Stockholm, and where I realized the power of data, especially genomics data, coming in not just in research, because we were coming from research community, we realized that a lot of data is coming in research. Genomics was coming in healthcare. There were these genomic tests available for various disorders, not just to identify or diagnose certain diseases, but also in preventive screening. For example, if we know that if we have a particular mutation which can be responsible for a disease at a later stage of time, that can be that can also be screened, can be used as screening test. We can use it for diagnostics. We can also use them for therapeutics, because if we know that there's a mutation in the gene which is making, so there's a very simple theory, maybe you have learned it in your schools, that gene get transcribes into RNA, DNA gets transcribed into RNA, RNA gets translates into protein. So if there's a problem in the gene, the protein gets a problem. Either the protein is not formed properly, different amino acid is formed, or there's some certain problem. If we can replace that particular protein using various therapies, or if we can provide a gene therapy, that the the disease can be cured to some certain extent. So basically, all that can be part of gene therapies. So with Genomic, we aimed at building such solutions, which were genome informatics solutions to design and develop, identify these mutations, and to look for various therapeutic approaches. So my talk today is about empowering medical decisions using various smart genome informatics solutions, so that we can start moving towards precision medicine. And to do that, we are making cloud-based AI-enabled solutions for analyzing and reporting genome testing data. So, so very simply, let me take you through the life cycle of a genomic test. When a doctor prescribes a genomic test to a patient, when a patient goes with a certain symptom, the patient sample is taken either in the form of blood or in the form of tissue for specific cancers, and the lab experiments get done. And to excite you more, a lot of data, GBs of complicated data, gets generated as a part of those sequencing experiments, which required efficient algorithms, efficient training to predict or identify those mutations, and trained bioinformaticians, genetic counselors. And I think this crowd definitely understands that there is a lack of many such skilled people, especially in our country and actually across the world as well, to identify the mutations and report the findings. Now, this is actually, I summed up everything in here. And if we actually look at the diagnostic reports, so somebody was asking, why did you take this startup journey? So when I was doing my postdoc in Stockholm, some of my friends had sent me certain data sets, the patient data, and to look into that data, because the reports that were provided to them were not matching or were not effective enough. When I reanalyzed the data, I realized that analysis was not done properly. So basically, the report that was given to the patients were wrong. So wrong reporting was being done in patients, which had not just has many other psychological or traumatic experiences for the patient, but also it's not good in the terms of medical community. And this leads to wrong decisions in terms of precision medicine as well. And the accuracy of report is not good because they are using manual approaches. Yeah, because in my next slides, I'll tell you that what exactly data comes up, how we analyze the data, and what exactly we do at the end. So it's a pipeline of certain 5 to 7 steps. And when we do this, go through this pipeline, we have to take care of accuracy or quality checks at each and every step. So, and when things are done manually, definitely there are certain problems that leads to certain errors. So that's why the reporting was very much prone to errors, and because it was done manually, the turnaround times still are very high in many labs. It ranges to weeks, where we are able to do it in a few hours at this point of time. So to solve these issues, and also by 2025, over 100 million genomes will get sequenced, and 20 billion GBs of just the raw data will get generated, which is in itself is a huge problem, because how are we going to analyze, interpret, make sense of this data, and also to manage and report the entire amount of data? So with all these problems, we realized that we need to come up with smart solutions, and we started making these cloud-enabled solutions. So let me take you through what happens when a sequencing is done. So the first is called NGS. NGS stands for next generation sequencing. It's just a simple process where an experiment is done to get the high throughput data from either your blood or your tissue. And when you do that, there are 3 levels of analysis, which includes a primary analysis, a secondary analysis, and a tertiary analysis. So, and after the tertiary analysis, what you get is a list of mutations that a person may have, and every other individual, we all are evolving. So we may have thousands of mutations in us, but what is that particular single mutation that will be responsible for that particular disease? To identify that, we have to do a lot of annotations and filtering and prioritization, use those steps. And finally, at the end, what we get are the actionable insights, that which particular mutation is responsible for this particular disorder. So I'm giving you actually as I said in the very beginning, that I'm giving you the idea of the applications wherein you can deploy your own algorithms and take through all these steps and identify, make classifiers to actually predict which particular variant can be very important, and how we can look for actionable insights in these from using this particular information. So using all this information, we were looking for variants in the, as I said, 3 billion base pairs. But actually what happens is, if I look at the size of genome, it's 3 billion base pairs, which is exactly like the size of this apple that is shown in the picture. And when we look at the protein coding genes, or the genes which are responsible to, or which might affect at least 85 to 90% of disorders, are just 20 to 25,000 genes, which are actually the size of this particular slice that is over there. So only, or in other terms, we have the information, or we have, right, till date, we have the knowledge to analyze or interpret or identify variants in that 1% part of human genome. We still are not very much sure that what happens in the rest 98, 99% of the human genome, which was, when I was in my master's or PhD, was called as the junk DNA. Now we know it's not the junk DNA, it's called, there are other names like non-coding part of the DNA or non-coding genome, but we still don't know how to make sense of that part. So still today, all of us, like all the genomic labs, are trying to make sense of that 1% part of the genome. And when we talk about cancer, and also because when we are sequencing the entire apple, it costs thousands of dollars, and if we try to sequence or do the testing of the smaller part, it is a bit cost effective. And since we are giving these tests to patients, we also have to take care of the costings as well. So, and when we talk about cancer, we even don't look at that 1% part of the genome. We look only for around maximum till 100 or 200 genes, which is like another 1% of that 1%. So that is the only information right now that we have out of the entire genomics after being studied in last 2 decades. But still, what we have achieved is, genomics as a community, that how these genes can affect, how the variations or mutations in these genes can affect or cause a particular disorder, and can we use this information to provide treatment modalities. Now, yeah, so when we get the data from a next generation sequencing or sequencing experiment for that matter, what we essentially get is these, what I have mentioned in the A segment, small segments, segments of stretches of reads, which is stretches of bases, which is like ATGC bases of small stretches. These stretches range from 100 nucleotides, 100 nucleotides or 100 letters to 200 letters or even more. Some of them are in MBs, but yeah, these are small stretches, and as shown in B, these stretches are supposed to be mapped to the reference genome. Now, Human Genome Project, which was completed in 2003, actually gave us a standard reference genome, which can be used as a reference set for all the humans of the world. Of course, there are certain drawbacks to that, because at that point of time, Indian genome was not there, and we could not add Indian information over there. Of course, so if we keep aside all the drawbacks, we still have a reference standard set of human reference genome, which is used to map the small stretches of sequence or the nucleotides that we get after the sequencing to the reference genome, so that we can finally, it is not, so that, okay. So that what we finally get, as in mentioned in the C segment, I think I can do from here. Yeah. So what we actually get is, once we map all these reads to the reference genome, all these reads as in the stretches of nucleotides on the reference genome, what we actually get is the information of where there is a difference in the nucleotides in the patient or the sample genome that we have as compared to the reference. And when we see, it's a very simple algorithm. So when we see, when there is a difference in the ATGC of the reference and in the patient as compared to the reference, we know that there is a mutation. And we right away don't call it a mutation, because then we have to score it according on the basis of the number of reads that are actually there. So we have to, where there are certain scoring methods. I don't have to explain it to this audience. You know much better than me how to do this scoring make these scoring algorithms. But there are scoring algorithms which define that, okay, for the particular mutation that we are seeing is actually there, or it's a noise, or there is an error over there. And then we compare it. If there is a patient sample, we also have a normal sample, so normal patient. So we compare it with the normal patient, and we identify that those mutations might already be there in the normal. So that mutation might be common in the population, but might not be having any particular disease-causing effect. But there might be certain mutations which will be specifically there in the tumor sample or the patient sample, which we then finally screen out from that data, and then we identify, okay, we prioritize that these are the variants which are different or specifically present in the patient info data, and are these variants, any of these variants, responsible for a particular disorder. So there can be different types of disorders. There can be monogenic, where the disorder might be dependent on one particular gene, polygenic, many genes, and so on and so forth. So basically, what we at the end of all this exercise get is the information of those variants which might be there in the patient sample and which might be responsible or causal factors for a particular disorder. And once we get those variants, and we call this step as called variant calling, we actually filter to prioritize and identify a causal variance based on the available literature, available information, and many other prediction algorithms as well, so that we can identify that this is a deleterious or this is a problem-causing or disease-causing variant. Now, all this information, where actually we use this information is used to identify or design precision medicine for a particular patient. So that we can tailor the medications on the basis of the genetic profile of an individual. So on the basis of genetic profile, we know that this is a patient with this particular disorder. We know that are these disorders inherited as in the disorder, or that these variants, if these variants have been passed through the family, I mean through the parents. There are parental screening also done, career screening is also done. We are also working on patients wherein we are identifying such inherited variants in the parents who, in the couples who are planning their children. So we do this is called career screening for those individuals who might be either having consanguineous marriages, and who might have history of miscarriages, where career screening is done on the basis of all this information to identify those mutations. Because what happens is, for us for any mutation to express or to show any effect on any individual, there should be 2 copies of those allele. This is called, we all have biallele, 2 copies of each allele should be there, and 1 copy will come from mother and the other copy will come from father. So if those 2 copies are there, then the mutation is going to cause any effect. So we also do these screenings. This is called career screening for individuals who are planning for the next child, and use all this information, that whether there is an inherited disorder, how we can use this information in precision oncology, how we can identify patients with certain cancer patients with disorders, and use all this information to provide them tailored medications. And I think I've covered all these things. So all this so that we can enhance the efficacy of the treatment. And I am reiterating most of these things. So yeah, and now this was mostly about the field, and now what we are also doing is, we do have a lot of information. We have worked with a lot of pediatric doctors. Our core field area of research is maternal and child care, wherein we are working on pediatric samples, rare inherited disorders which are there in kids. We are working on those children. Then we are also working on children having solid tumors, whether those solid tumors are inherited, what is the inheritance pattern. So we are also screening the parents and the kids for that. And if are they also acquiring these mutations in such a small period of time. The broader idea or the vision is to develop newborn screening panels for these kids using this particular information. And herein we are also including a lot of pharmacogenomic information. Now, when I say pharmacogenomics, what exactly it is? So now I've been talking about mutation. There is one concept called mutation. There is one concept that, okay, there are certain drugs that can be applied for that particular disorder, which like for that particular mutation, targeting that particular mutation. But does that drug will that drug work on me, or will that drug work on you? Because there have been studies which show that there are specific drugs which work according to the genetic profile of a particular individual. So, and this particular field, which is called pharmacogenomics, basically tailored medications on the basis of the genetic profile, can we use this genetic profile of the patient to identify these causal variants and optimize the selection and efficacy of the drug therapy. So now, so our first part wherein we are identifying or making the diagnostic reports is kind of done, and we are having clients for that. We are working on the second aim to identify the optimization of and selection of the drug efficacy using various pharmacogenomics data. And we are trying to build AI-enabled models for that. And as I have told to Karthik, we are a bioinformatics company, and we will definitely look for collaborations from people like you to help us develop such modules so that we can collaboratively build solutions for our society. Now, this was the kind of cloud-based platform that we started with. So this is actually the solution that we talk about, wherein the raw data from the users, which actually include the raw data, the various genome testing data, and our users are the hospitals, diagnostic labs, various doctors, will go through the pipeline, that the algorithm that we have made, and will provide various detailed information about the genomic biomarkers, the pharmacogenomic results, and various other the customizations will be provided on the using that particular data. So these are the different solutions that we have right now. So the first solution is called OnCare, where we are working on targeted cancer panels, which are also known as onco panels, and providing the genetic information, basically the actionable insights which can be used by doctors to provide therapy recommendations. The second is Anuvanshiki, and as the name suggests, we are looking for inherited disorders. We are working on inherited disorders here and identifying the specific mutations related to hereditary disorders. And then we are also working on various customized solutions in this particular area. These are the different screenshots of our platform, which is currently under development, but yeah, we provide a secure dashboard to a hospital, and wherein they upload the raw data set that they get from the sequencing company, and then the final report is provided to them through their dashboard. And these are the different informations, the variant information, various other information that are provided to them, which is required by a doctor to identify, analyze, interpret, and finally report the results to a particular patient. These are the different types of information is provided in our reports. In the first column, as you see here, what we provide is that actual genomic alteration present in the patient. EGFR is a very well-known actionable marker. It's a gene actually. It's a gene, and what amino acid changed to what amino acid at what particular position. So L changed to R at 858 position. This is the type of accuracy we have to provide in the report. And are there any and then what are the FDA approved therapies that can be used for this particular if that particular mutation is there, and what are the different clinical trials that are ongoing for this particular can be provided or ongoing for this particular mutation. And then different details of those therapies are there, that of this mutation is there, that what is the frequency, what is the scorings, and what is the classification level, and we follow a lot of guidelines provided by various guidelines programs. Now, we have been working with a lot of hospitals and diagnostic labs of the country, and we collaborated with them to work on various samples. The sample list size is not very big, but yes, we are still in the stage of making more collaboration so that we can increase the sample size and report the validations and accuracy and efficacy maybe in my next talk whenever I'm here. These are the different clients that we have been working with. We have, apart from these solutions, we have been also providing various dedicated genome informatics services. So these were the solutions that we were making, especially on the patient data sets. And then we were also working on some research projects with some research institutes. And these were the different people whom we have delivered certain projects. The last but not the least, I would like to introduce my team. Sunil is a scientific advisor and was the founder of Indian Centogene, and now he's with us. And then Saurabh and Padmini are my both hands who are taking care of all the entire bioinformatics. And then we have a lot of consultants and other advisors who are with us as a part of our team. And this is it from my side. And before ending, as speaker before me, Manas, was talking about his experience, I would like to share. I don't have slides for that, but I can, speak if I have time. Can you have some time up? She is going like this, time is up. But I'll take just 5 minutes. So, okay, so actually, though I have spent a long time in academia, almost 15, 17 years, published in studied in various Indian or international labs, published high quality papers, and, roamed across the world. But what I realized is that if you don't, use your skill or your, knowledge to build something for your community or society, this was the trigger point for me that if I could continue working in the same academic system throughout my career, but if I had that knowledge or power to build something for the society, I think I can make a small change. And if I can motivate any one of you to start working on your, building small things, for not just for yourself, but for your community, for your society, for patients, or for general, community, it will be really helpful. Because what we are fortunate enough to reach at this stage, to have that family who supported us to study, to have that community who supported our careers, to have that knowledge that has brought us here. We were fortunate enough to do that. But there are people out there who don't have that, a privilege. So if we have that privilege to stand and talk to such an audience, we can actually motivate people to, build things and solutions. Not just there are different types of solutions, and to take that plunge out of their careers to start building for the nation. That's something that I would like to end my talk with. Thank you for your time and attention. I'm open for questions. And if you want to reach out to me, please follow that QR code. By the way, I'm looking for a CTO. So, okay.